Okay, so in continuation with what I was discussing about the vectors, these TA plasmid vectors, they have some limitations and also plus points. There is vectors which are derived from the TA plasmids. The binary vectors which have been derived from the TA plasmids, relatively they are of large size, and very less number of restriction sites were available with this particular type of plasmid, which reduced the capacity of the vector system to carry the DNA. So for that, in order to enhance, people have derived one more vector called as PB19. It was predicted that more than half of the DNA could be deleted and the vector would still be completely functional. So that's why instead of 9.8 kilobase pairs of original vector, now they have developed a mini binary vector of 3.5 kilobase pairs of PCB301 was constructed, which increased the capacity of insert into the binary vector systems. This mini vector, which can be used for cloning in fragments of DNA and can be infused into the tumefactions by conjugation, because certain regions of DNA required for conjugal transfers has been deleted, but they could able to transfer this particular DNA into the tumefactions through electroporation and then achieve the transfer into the plant DNA. So then after this, again people have actually, in order to solve the problems of TA, because it's found that TA, the agrobacterium tumefactions doesn't infect all kinds of plant cells. So then in order to solve this, people started looking about other possible bacterial systems which actually infect the plant cell. So then they found another type of agrobacterium species which was involved in the infection into the plant cell and carries the plasmid structures similar to that of TA. So the organism is Agrobacterium rhizogenes, which is a gram-negative soil bacteria which is responsible for hairy root disease in case of dicotyledonous plants. And in rhizogenes induces the formation of proliferation of multi-branched adventitious roots at the site of infection, so they call it as hairy root. So rhizogenes is a causative agent, and it can cause this particular kind of problem because of presence of plasmid called RI plasmid, that is hairy root inducing rhizogene infecting one. So here in this particular RI plasmid, it was found that it's a large plasmid carrying around 200 to more than around 800 kilobase pairs of DNA, and contains one or two regions of tDNA and a virulence region and all the other kind of genes necessary for causing the hairy root formation. So RI plasmid are grouped into two main classes. One class is called opine synthesizing class, which causes synthesized by hairy root, and first is agropine type and another one is monopine type. So agropine type strain introduces the roots into the synthesizes agropine and monopine and related acids. Second is monopine type strain induces the roots to produce monopine and the corresponding acids. The agropine type RI plasmids are very similar as a group and quite distinct group from the monopine type plasmids. So this is the structure of this particular plasmid, the RI plasmid. So this is a vector which was derived out of the RI plasmid, carrying origin of replication of E. coli, followed by origin of replication of rhizogenes, then non-terminated regions, NPT for selection marker, and had multiple cloning sites, 35S promoter of cauliflower, then right border, left border region. So this particular was a tDNA construct which was used for the transfer of the foreign DNA into the plant system. Then the genes responsible for the hair root formation was found to be present within the tDNA. So means they are present in the plasmid. The tDNA of the agripine type RI plasmid consisting of two separate tDNA regions designated as TL and TR. So that is having about 15 to 20 kilobase pairs, and these two fragments can be transferred independently during the process of infection. The genes encoding for auxins synthesizes the TMS1 and TMS2, and agripine synthesizing AGS has been localized into the tRDNA. And monopine RI plasmids contain only one tDNA, which is considerable DNA sequences homology to the TI plasmids. So this is the role A, role B, role C are the most important genes which is responsible for the hairy root induction. Role A associated with the internode shortening, leaf wrinkling and other things. Role B is responsible for protruding the stigmas and reducing the length of the stamens. Role C causes the internode shortening and reduces the apical dominance. So although tRDNA is not essential for the hair root formation, it is found that AXU1 gene harbored in this particular segment responsible for that. So this is how the transfer of gene takes place between the agrobacterium and the plant cell. You can see the virulence genes are going to help in excising the single fragment of the T strands, and then the single stranded tDNA complexes with the virulent protein molecule, and then one of the virulent protein will be going to form the channel through which the protein complexed DNA is going to get transferred into the plant host system, plant cell, and within the plant cell it will also get carried. It will not leave that particular completely the DNA as such within the cytoplasm. So there will be a number of other proteins which will again interact and carries it to the DNA. So this is also a method of transfer of DNA molecule biologically into the plant system. So you can see here the plant cell take a agrobacterium DNA, means agrobacterium, and inserts the, generally this is how actually it is going to be inducing. So generally in order to do the transformation, people actually collect the cell from the plant, and the plant cells are going to get transformed by using the bacteria which are actually having the foreign DNA molecule, and then those particular cells which get integrated with the foreign DNA will be selected onto the medium having the marker molecule, and then those selected cells are going to be subjected for multiplication. So this is how the transfer of DNA is going to be done into the plant cells. Now in order to identify whether the gene of interest has got transferred into the plant system or not, people use some reporter genes, means in order to say whether our target protein is being synthesized or not, or in order to suggest that whether that particular transfer is achieved properly or not. Okay. So the reporter genes, these are the genes which are going to help us in understanding the synthesis as well as to know whether the gene construct has got successfully transformed into the plant cell or not. So some of the reporter genes are the GUS-A, that is beta glucuronidase gene. So this beta glucuronidase gene is sourced from the E. coli and involves in the catalysis and hydrolysis of beta glucuronidase. The assay is going to be non-isotropic, and in vitro assay are going to be through colorimetry or fluorometry. It can be done, and it is in combination with a X molecule or X group in the form of X group. Advantage wise, it is very simple, sensitive, quantitative method, and it is disadvantage wise, it's a destructive method, and the enzyme is stable, so unsuitable for the studies of down regulation. Then there is CAT gene, there is chloramphenicol acetyl transferase gene. So again, it was taken from E. coli, and activity wise catalyzes the acetyl transfer and inactivates the chloramphenicol and gives an resistance to the chloramphenicol. And assay in vitro, and it's only isotopic assays can be possible. Advantage wise simple, and disadvantage wise low sensitivity and expensive, low resolution in vivo. Another is luciferase gene. So from the firefly, then activity wise light produces in the presence of luciferase. It's stable luciferin, oxygen, magnesium and ATP are required. Assay wise non-isotropic, bioluminescent assay can be done in vitro and in vivo. Advantage wise sensitive, rapid turnover is there. It can be used for quantitative estimations also. Disadvantage wise, it's very expensive. Detecting instruments are going to be needed, and limited reproducibility is seen. There is anthocyanin regulatory genes also being used from zeamides. So it induces the pigment and assays visual screening, and advantage is simple, inexpensive, non-destructive, but disadvantage wise low sensitivity. Then people gone into the GFP, green fluorescent protein molecule, so also used as one of the reporter genes. So these are again intrinsic fluorescence is going to be there under blue light, associated from the jellyfish, and assay wise non-isotropic, and in vivo can be done in a plant. So advantages wise intrinsic activity will be there, sensitive, even with the live plant we can do the assays. And then disadvantages wise weak signals in some systems, wherein GFP will not be able to give fluorescence properly, but it can be addressed by doing some small modifications within the GFP gene. So these are all few reporter proteins, genes which have been used in case of plant system for the process of means to express the foreign DNA into the plant system. So how to transfer? This particular transfer is being achieved by using protoplast transformation. So two procedures are there commonly used. One through protoplast, including naked DNA. It can through PEG it can be made to get acquire it. Our plant protoplast can also be transformed by using agrobacterium. So followed by transformation, the protoplast can be placed into the suitable selective medium, so which will regenerate the new cell, and plants can start multiplying and develops into a callus. Other methods of transformation, people have used particle bombardment method, microinjectile methods. So wherein the DNA is going to be coated around to the small particle, gold or small glass bead or silver beads, which are not going to be reactive, and then these particular particles are going to be bombarded onto the plant tissue material, and those bombarded tissue material will be further allowed to multiply and selected for the presence of the marker and reported genes, and then it can be amplified and developed into a plantlet. So these are all few things about how exactly transformation is achieved in case of plants. Then further we will enter into the specific type of promoters which have been used in plant system and applications of the plant biotechnology, which we will see in the next videos. Thank you.